Usually when we talk about getting a medical diagnosis, there's this very comforting expectation of precision.
Right. It feels like engineering.
Exactly. Like if you take a hard fall and break your arm, you go to the hospital, they throw you in an X-ray machine, and a few minutes later there's a film up on a light board.
Yeah, and the doctor just points to a jagged white line across the bone and says, well, there's the problem.
Right. And that binary nature of structural medicine is just incredibly reassuring. I mean, a bone is broken or it isn't.
We crave that visibility as humans. We desperately want to categorize our pain and, physically see the mechanical failure with our own eyes.
But the thing is, the moment you step out of orthopedics and into the world of the human mind, into psychology and behavioral science, that X-ray machine suddenly feels, well, completely useless.
Yeah, the landscape gets really murky.
It gets subjective and just infinitely more complex. But here is the hook, and this is where the sources for our deep dive today start to paint this fascinating picture.
What if the mind is just as mechanical as that broken arm?
Exactly. What if it's just operating on a scale that we struggle to comprehend? So let me throw a story at you from the sources.
Sure, lay it on me.
So the brilliant neurologist Oliver Sacks documented this case of a young medical student.
Yeah.
The literature calls him Stephen D.
Right, I know this one.
Yeah. So Stephen was, at the time, a pretty heavy recreational user of cocaine, PCP, and amphetamines. And one night, while under the influence, he falls asleep and has this incredibly vivid, visceral dream that he is a dog.
I mean, given that cocktail of synthetic stimulants and hallucinogens, a bizarre dream is probably, the least remarkable outcome he could have expected.
That's true. But the bizarre part wasn't actually the dream itself. It was the hangover.
The hyperosmia.
Right. Stephen woke up the next morning and discovered that his sensory perception had just been completely hijacked. He had this condition, hyperosmia, where his sense of smell had intensified to this superhuman or really canine degree.
He essentially woke up in the olfactory world of a golden retriever.
Literally. He walked into a clinic and realized he could instantly identify all of his friends and colleagues just by their unique body odors.
Wow.
Yeah, he could navigate his hometown blindly, purely based on the localized smells like the butcher shop, or the damp brick of a specific corner, or the exhaust of passing cars.
So he's experiencing the environment through a sensory channel that, well, humans typically relegate to the deep background.
Right, but it wasn't just a cool party trick, which is what makes this case so vital for what we're talking about today. This sudden shift in his physical sensory wiring completely overwrote his emotional and cognitive reality.
It altered his baseline.
Yeah, Sachs noted that Stephen felt this intense, unexplainable yearning, like a melancholia mixed with this overwhelming desire to physically smell and touch everything in his path.
Which is very dog-like.
Exactly. And even more unsettling for a medical student, his ability to think abstractly just collapsed.
Wait, really?
Yeah, he couldn't grasp complex, high-level theories anymore. He was so fiercely anchored to the immediate, overwhelming sensory experience of the physical world that philosophical reflection became nearly impossible.
Because the raw data stream from his nose was demanding so much processing power, right? The higher level functions of his brain were essentially starved of resources.
That's exactly it. Now, exactly 3 weeks later, the hyperosmia vanished as suddenly as it appeared. He just went back to normal.
Leaving him with just the memory of this intensely different version of himself.
Right. But looking at Stephen's case forces a really uncomfortable question. I mean, if a temporary chemical alteration in our sensory hardware can completely erase our ability to think abstractly
and rewrite our emotional baseline.
Yeah, then what is the self? Like, how much of what we consider our personality is just a fragile byproduct of our physical wiring?
And that underlying anxiety is the perfect engine for our mission today, because our goal for this deep dive is to explore the hard biological foundations of psychology.
Right. We're synthesizing a massive stack of research, neurological case studies, and evolutionary biology to prove a single point to you, the listener.
Everything that makes you, well, you. Your spontaneous thoughts, your sense of awe looking at a starry sky, your moral outrage, even your perception of the audio playing in your ears right now.
It's all rooted in the physical, tangible architecture of your nervous system.
We are essentially opening up the user manual for the human mind.
And historically, we have fought tooth and nail against this idea. I mean, you look at René Descartes, the famous French philosopher.
Right, the 17th century mind-body dualism.
Exactly. Descartes looked at the anatomical science of his era and realized the body was a biological, machine. He saw this system of hydraulic tubes and switching gears.
But he looked at the brilliance of human thought and decided that reducing our entire existence to, wet machinery was just unacceptable.
It would destroy the magic. And frankly, it would get him in a lot of trouble with the church back then. So he proposed a compromise. He argued that an immaterial, ethereal soul sits somewhere inside this slushy machine.
Pulling the levers, making the actual choices, and giving us free will. Descartes felt this intense need to build a firewall around the human experience.
Because the idea that something as sweeping as romantic love or as debilitating as grief could emerge purely from physical matter felt reductive to him.
Right. But modern neuroscience paints a picture that is infinitely more fascinating than simple tubes and gears. The reality is that we are unimaginably complex, deeply interconnected electrochemical networks.
And realizing that doesn't reduce the poetry of the human experience, does it?
Not at all. It actually grounds it and makes it more miraculous.
Right.
But to comprehend how this current network operates, we have to recognize that nature didn't design the human brain in a sterile lab with a master plan.
No, it was cobbled together blindly over millions of years of desperate survival. Nature is the ultimate, unapologetic recycler.
Which introduces a concept from evolutionary biology that runs through our entire stack of sources pre-adaptation.
Coined by the biologist Ernst Mayr, right?
Yes. The core idea is that evolution rarely invents a brand new biological system from scratch when an organism faces a new problem.
It takes an existing system that was built for one specific purpose, and it hijacks it. It co-opts it for a completely new function.
The classic anatomical example is the human mouth. The jaw, the teeth, the tongue. These evolved purely for the mechanical breakdown of food.
But millions of years later, when early hominids needed to communicate complex ideas, nature didn't grow a second organ on our foreheads for speech.
Thank goodness.
Right. It just hijacked the eating apparatus to articulate sound. The hardware was already there, so evolution just updated the software to give it a secondary function.
And applying this concept of pre-adaptation to our psychology unlocks some massive mysteries about human behavior. Take the concept of disgust.
Oh yeah, Paul Rosin's research.
Right, the American psychologist who literally goes by the moniker Dr. Disgust. He formulated this brilliant theory about how moral judgment evolved from physical survival.
Let's trace that lineage for the listener. Imagine you are a primitive human, and you stumble across a rotting animal carcass. You lean in and smell the rotting meat. What is your immediate, involuntary, physical reaction?
I physically recoil. My nose scrunches up tightly to block the odor.
Your mouth drops open.
Yeah, my tongue might thrust forward slightly in a gag reflex. I start salivating heavily, and my stomach churns with intense nausea.
Charles Darwin actually wrote extensively about this specific physical sequence. That facial contortion, the gape, the nose wrinkle is a highly evolved, localized survival mechanism.
It's designed to physically prevent pathogens from entering your respiratory system and to forcibly expel any poisonous material you might have just put in your mouth.
The nausea is your stomach preparing to reject toxic food. It's a biological alarm system dedicated purely to keeping your physical body from being poisoned.
But Rosin argues that as humans evolved into highly complex social creatures, we faced a new kind of threat. We weren't just threatened by poisonous food anymore.
We were threatened by poisonous social behavior.
Exactly. Cheaters, murderers, individuals who violated the social contract threatened the survival of the group.
Now, I think I see where this is going, and I want to push back on it a little because it sounds almost too neat.
Okay, go ahead.
Are you telling me that when I read a news article about a corrupt politician stealing millions, or I hear about a horrific war crime, and I say, that makes me sick to my stomach, it's not just a linguistic metaphor?
It is not just a metaphor.
My brain is literally processing a complex ethical violation the same way it processes a piece of moldy bread.
The clinical data backs that up entirely.
Yeah.
When researchers place subjects in an fMRI machine and ask them to evaluate severe moral offenses, the scans show massive activation in the basal ganglia and the right prefrontal cortex. Those are the exact same neural networks that light up when you expose a subject to the smell of rotting food or show them a picture of feces.
Wow.
Evolution needed a way to make us recoil from bad social actors, to ostracize them. It didn't have the time or the biological budget to build a brand new moral outrage center in the brain.
So it's simply routed our complex, newly evolved social evaluations through the ancient, pre-existing, don't eat that poison circuitry.
Exactly. It's wildly efficient.
That reframes the entire concept of morality for me. What we consider morally acceptable is obviously shaped by our culture, but the physical engine that makes us feel the violation of those morals is just a repurposed vomit reflex.
Yeah. And there is an even darker example of this hijacking in the research regarding social exclusion.
This is the DeWall and Baumeister experiment.
Right, because humans only survived the African savanna by banding together. Being isolated from the tribe meant you were going to be eaten or starved to death. Social isolation was a literal death sentence.
So to study how our modern brains handle this ancient threat, DeWall and Baumeister designed an experiment that, frankly, borders on psychological torture.
It is a landmark study, but yes, the methodology is intentionally brutal to elicit a genuine biological response.
So they brought subjects into the lab and had them complete a comprehensive personality assessment. But the results they handed back to the subjects were entirely fabricated.
They split the group in half. The first group was told that their profile indicated a rich life full of stable, lasting relationships.
But the second group was handed a devastating prognosis. They were told, in this clinical, authoritative language, that they were fundamentally incompatible with other people, that their future relationships were guaranteed to fail, and they would end up completely alone in life.
They induced a state of profound existential social rejection in that second group. They made them feel entirely ostracized.
And then, the researchers subjected both groups to a physical pain tolerance test. And this is where my intuition completely failed me reading the source material. Well, you would naturally assume that someone who has just been emotionally crushed, someone who is feeling devastated and fragile, would be far more sensitive to physical pain.
Sure, that makes intuitive sense.
But the data showed the exact opposite. The people who were told they were destined to die alone actually exhibited a significantly higher tolerance for physical pain. They had become physically numb.
Right.
I struggled to understand the logic behind that.
Well, the logic only reveals itself when you view it through the lens of pre-adaptation. Consider how a wild animal responds to a massive physical trauma.
Okay.
If a zebra has its leg torn open by a lion, it cannot afford to be paralyzed by the agonizing pain in that moment. If it stops to process the pain, it dies.
So it runs.
Exactly. The biological response to severe injury involves releasing a flood of endogenous opioids, natural painkillers, to temporarily numb the body, raising the pain threshold so the animal can escape.
Oh, well, I see it now.
Apply that to the early human. Being kicked out of the tribe was just as lethal as having your leg torn open. It was a mortal threat. So nature frugally wired the distress of social rejection directly into the physical pain system.
So when you, the listener, go through a devastating romantic breakup, and you say you feel numb, or you describe the loss as feeling like a physical blow to the chest, your nervous system isn't differentiating between emotional heartbreak and getting hit by a car.
Physiologically, they are intertwined. The emotional numbness that ostracized people report is a defensive survival mechanism. The brain perceives the social rejection as a physical injury and deploys the same painkillers to help the organism survive the immediate shock.
That's just a stunning display of biological efficiency.
Neuroimaging confirms it too. The emotional pain of being left out of a group activates the anterior cingulate cortex, a region heavily involved in processing the distress of physical pain.
So to truly understand how a breakup causes physical numbness, or how a moral failure causes nausea, we need to physically map out the hardware that carries these signals.
Right, we have to look at the grand architecture of the nervous system,
which our sources define as every single piece of neural tissue in the human body. And this massive network is divided into two primary domains: the central nervous system, or CNS, and the peripheral nervous system, or PNS.
And the easiest way to visualize this division is by looking at the physical security of the tissue.
Security.
Yeah. The central nervous system is the most vital, delicate equipment you own, so it is heavily armored. It consists of the brain, securely encased in the thick bone of the skull, and the spinal cord running down the center of your vertebrae. That is the command center.
Got it.
Everything else, all the neural tissue extending outward from that central column to your fingertips, down to your toes, wrapping around your internal organs, makes up the peripheral nervous system.
I look at it like the architecture of a massive modern smart home. The central nervous system, the brain and spinal cord, is the primary server rack sitting behind a locked door in the basement.
Running the complex algorithms, storing the memory.
Right. But a server in a basement is entirely useless if it can't interact with the house. So the peripheral nervous system acts as the miles and miles of physical fiber optic cables running inside the drywall, connecting that isolated server to every single smart device, thermostat, and security camera in the building.
That's a great analogy.
And those peripheral cables have to run two-way traffic. You have afferent nerves, which are the sensory pathways carrying information from the body inward to the brain, like a thermometer sending a temperature reading to the server.
And you have efferent nerves, which carry the motor commands from the brain outward to the body, like the server telling the air conditioning unit to kick on.
And if we look even closer at the peripheral nervous system, the cabling outside the brain, we find it's further divided into two distinct subsystems.
Based on the level of conscious control we have over them. The first is the somatic nervous system. This system interacts strictly with the outside world,
Carrying sensory input from the skin and joints.
Exactly. It carries that input up to the brain, and it carries motor commands back down to those skeletal muscles. This is the domain of voluntary conscious action.
So when you feel a mosquito land on your arm and you consciously decide to raise your other hand and swat it, you're using the somatic network.
Exactly. But the body has an endless list of chores that I absolutely do not want to be consciously responsible for.
Oh, same. I don't want to have to manually remind my heart to beat 80 times a minute or consciously contract the muscles in my stomach to digest my breakfast.
Boy, if you had to think about that, you'd forget to breathe the moment you got distracted by a text message.
Which is why nature developed the second peripheral subsystem, the autonomic nervous system. Autonomic shares a root with autonomous, meaning self-governing. It's the biological autopilot.
It controls your internal organs, your glands, and your smooth muscles. It manages your blood pressure, your pupil dilation, your digestion. It's the silent background operator ensuring the machinery stays alive while your conscious mind is busy trying to solve a crossword puzzle or listen to a deep dive.
Okay, so we have the armored server rack of the CNS and the vast cabling of the PNS. But if we pull back the drywall of the body and put a piece of that cabling under a powerful microscope, what are we actually looking at?
You're looking at the fundamental unit, the basic building block of this entire global network, the neuron.
And a neuron is a highly specialized biological cell designed for one singular purpose.
Right.
The rapid transmission of information.
Yes. They come in an astonishing variety of shapes and sizes depending on their location, but they all share a core anatomical blueprint.
Let's break that blueprint down.
In the center, you have the soma, which is the cell body. It contains the nucleus and the biological machinery that keeps the cell alive. Radiating outward from the soma are multiple branch-like structures called dendrites.
The word dendrite actually comes from the Greek word for tree, doesn't it?
It does, which perfectly describes their branching canopy-like appearance.
And those dendrites are the designated receivers. They act like a network of little antennas constantly listening for signals from other neighboring neurons.
Precisely. They collect incoming chemical messages. If the soma receives a strong enough signal from its dendrites, it decides to pass the message along.
It fires an impulse.
Down a single elongated fiber called the axon. The axon is the transmission line carrying the signal away from the cell body. And at the very end of this line, the axon branches out into multiple tiny swellings known as terminal buttons.
And those buttons are responsible for handing the signal off to the dendrites of the next neuron in the chain. What always stuns me is the physical scale of these cells.
It is wild.
A neuron packed into the cerebral cortex of your brain might be entirely microscopic, its axon stretching only a tiny fraction of a millimeter to talk to its neighbor. But a sensory neuron located in the base of your spinal cord might have an axon that runs all the way down your leg to the tip of your big toe.
That is a single, continuous biological cell that is over a meter long. It is an incredible feat of biological engineering.
And we classify these varied neurons by the direction they carry information. Sensory neurons are the afferent pathways bringing data inward. Motor neurons are the efferent pathways carrying commands outward.
And sitting entirely within the brain and spinal cord are the interneurons. They are the middlemen, receiving sensory data, processing it, and routing it to the appropriate motor outputs.
Now, before we get into how these neurons actually talk to each other, we have to address a massive piece of pop culture mythology.
Oh, I know what you're going to say.
We have all heard the statistic, repeated endlessly in movies and self-help seminars, that human beings only use 10% of their brains. The pitch is always, imagine the genius you could unlock if you could just tap into the other 90%.
It is a pervasive, completely unfounded myth. Brain imaging clearly shows that we use 100% of our brains. There are no dormant, unused sections just waiting to be unlocked.
But the origin of this myth likely stems from a very real early misunderstanding of brain tissue composition.
Right. It is a physiological fact that neurons only account for a fraction of the total cells in the brain. The vast majority of the cells in your skull, making up more than half the physical volume of the brain, are non-neural cells called glial cells.
And the scientific community did the glial cells dirty right from the start. The word glial comes from the Greek word for glue. For decades, early neuroscientists assumed these cells were just biological packing peanuts.
They thought they were just dumb structural putty holding the very important neurons in place so they didn't slosh around in the skull.
But that perspective is radically outdated, isn't it?
Completely. It's honestly dismissive of their crucial role. Glial cells are incredibly dynamic and essential to survival. They act as the brain's housekeeping staff, constantly scavenging and removing metabolic waste products.
And during embryonic development, they do even more.
Right. Specialized glial cells form the physical scaffolding. They create literal tracks that newly born neurons migrate along to find their precise geographic location in the developing brain.
They provide nutrients, form the brain's immune system, and modern research shows they actively participate in regulating neural signals. So if the neurons are the divas standing in the spotlight on stage, the glial cells are the massive, indispensable stage crew managing the lights, the sound, and the rigging.
Without them, the show stops immediately. I love that analogy.
But let's put the spotlight back on the divas for a moment, because I have a mechanical problem with that meter-long axon we talked about.
Okay, what's the problem?
If I step on a sharp piece of glass, a sensory neuron in my foot has to send a pain signal a full meter up my leg to my spinal cord. If that axon functioned like a standard copper electrical wire, the natural electrical resistance of the biological tissue would cause this signal to degrade and fade away before it ever reached my knee.
That's true.
So how does a biological cell transmit a signal over that vast distance without losing strength?
This is where we have to abandon the copper wire analogy. Neurons do not passively conduct a continuous flow of electricity like the cord plugged into your wall. Neural communication is an active, dynamic electrochemical process known as the action potential.
The action potential.
Yes. To understand it, you first have to understand that the outer skin of the neuron, the cell membrane, is not just a dumb wrapper. It is a highly sophisticated, semi-permeable barrier. Embedded throughout this membrane are microscopic pores called ion channels.
And these channels operate like highly secure, selective doors that only allow specific electrically charged molecules, known as ions, to pass in and out of the cell.
And the major players in this electrical bands are sodium ions, which carry a positive charge, potassium ions, which also carry a positive charge, and chloride ions, which carry a negative charge.
Okay, so imagine a neuron that is at rest. It's not currently sending a signal, but it isn't asleep. It is actively preparing to fire.
Using specialized mechanisms called ion pumps, the cell constantly burns energy to force positive sodium ions outside the membrane while actively pulling potassium ions inside.
Because of how these ions are distributed, along with negatively charged proteins inside the cell, the interior of the neuron ends up with a net negative electrical charge compared to the fluid surrounding it outside.
Right. If you insert a microscopic electrode into a resting neuron, you'll find it sits at a resting potential of approximately negative 70 millivolts. It acts exactly like a tiny, fully charged battery.
It's electrically polarized. It's holding this immense electrochemical tension, just cocked and waiting for a reason to fire.
It is sitting on a hair trigger. As this resting neuron receives chemical signals from its neighbors at its dendrites, those signals subtly alter the voltage. They might nudge it from negative 70 up to negative 65 millivolts, slightly depolarizing it.
But the ion pumps easily clean that up and push back down to negative 70.
Usually, yes. However, if the incoming signals are strong enough and they push that internal voltage up to a critical mathematical tipping point, usually around negative 55 millivolts, everything changes instantly.
That negative 55 millivolt mark is the excitation threshold. The second the internal voltage hits that number, the cell membrane suddenly becomes highly unstable. Specialized voltage-gated sodium channels slam wide open.
Because the inside of the cell is negatively charged and there's a massive concentration of positive sodium ions trapped outside, basic physics takes over. Opposite charges attract.
The sodium floods violently into the axon. In a fraction of a millisecond, the internal voltage spikes from negative 55 all the way up to positive 40 millivolt. That massive, temporary, localized reversal of the electrical charge is the action potential. It's the spark.
And just as quickly as it peaks, it resolves. The sodium channels slam shut, potassium channels open to let positive charge flow out, and the ion pumps aggressively go to work resetting that negative 70 millivolt battery.
This entire spike and reset takes mere milliseconds.
And this spark doesn't just happen once, it triggers a chain reaction. The depolarization of one segment of the axon triggers the sodium channels in the adjacent segment to open, and the spark travels down the entire length of the axon like a flame burning down a
So it
fuse.
never loses strength because it is constantly being regenerated at every point along the membrane.
Exactly.
This precise mechanism is so fundamental to our existence that if you interfere with it, you can completely erase reality. Which brings us to the dreaded dentist chair.
Oh, Novocaine.
Right. If you need a cavity filled, the dentist injects a local anesthetic like Novocaine into your gum. But what is that drug actually doing on a biological level? It doesn't dull the drill, and it doesn't change anything in your brain.
Novocaine is an elegant, targeted chemical weapon against the action potential. The drug molecules physically wedge themselves into the sodium channels of the sensory neurons in your jaw. It acts exactly like a cork jammed tightly into a bottle.
So when the dentist starts drilling, the physical trauma causes the neuron to hit that negative 55 millivolt threshold. The cell tries to open its doors to let the sodium rush in to create the pain signal.
But because the Novocaine is corking the channels, the sodium is blocked. The cell can never reach the positive 40 millivolt spike. The action potential is fundamentally physically impossible.
So the drill is still tearing through the tissue. The physical damage is absolutely occurring. But because the electrical spark can't ignite, the message literally never makes it past your jawline. Your brain is sitting in the dark, blissfully unaware of the destruction happening an inch below it.
It is mind-blowing.
It really is. But I want to go back to the speed of this process. Even with the action potential regenerating down the line, having to open and close millions of tiny ion channels millimeter by millimeter down a meter-long leg nerve sounds horribly slow.
It would be.
If a lion bites my foot and the signal takes 2 seconds to reach my brain, I'm lunch.
Nature recognized the speed limit and engineered a brilliant physiological upgrade. Many of the long-distance axons in your body are wrapped in a protective casing called the myelin sheath.
Remember those vital glial cells?
Yes. Specialized glial cells physically flatten themselves out and wrap tightly around the axon like layers of electrical tape, providing dense insulation. But crucially, they don't cover the entire axon continuously. They leave microscopic, uninsulated gaps between each wrapping,
Which anatomists call the nodes of Ranvier. So instead of the action potential having to painstakingly burn its way through every single consecutive millimeter of the membrane, the myelin insulation forces the electrical charge to literally leap from gap to gap.
It jumps from one node of Ranvier to the next. The Latin word for leap is saltare, so this process is called saltatory conduction. This leaping mechanism massively turbocharges the speed of the signal, rocketing it up to 120 meters per second.
And we can graphically see how vital this insulation is by looking at what happens when it degrades. Multiple sclerosis, or MS, is a devastating autoimmune disease where the body's immune system gets confused and actively attacks and destroys the myelin sheath around axons.
Without that thick insulation, the electrical signals leak out. They short circuit, slow down dramatically, or fail to reach their destination entirely. This loss of signal integrity leads to the severe sensory distortions and progressive loss of motor control that characterize the disease.
So assuming a healthy neuron, this electrical spark leaps rapidly down the axon. And here is where we hit a fascinating paradox regarding how we experience the world. The action potential is governed by what is called the all or none law.
It operates exactly like the trigger of a gun. You cannot half-pull a trigger to shoot a bullet slowly.
Right. Once you apply enough pressure to break the threshold, the mechanism fires and the bullet exits at the exact same velocity every single time. Neurons are binary. They are either at rest or they fire a full positive 40 millivolt action potential. There's no such thing as a small action potential or a large one. Every single spark is identical in size.
They are the fundamental ones and zeros of your biological computer.
But wait, if the electrical signal traveling up my arm is the exact same size, whether a feather touches my skin or someone hits my arm with a hammer, how does my brain know the difference? If the ones and zeros are identical, how does the system encode the intensity of the physical world?
That question strikes at the heart of neural coding, how simple binary signals represent infinite complexity. The nervous system relies on two primary strategies to translate intensity. The first is frequency coding.
Frequency coding.
Even though the size of the electrical spike never changes, the rate at which the neuron fires can vary wildly. A feather gently brushing your arm might cause a sensory neuron to fire a leisurely 20 action potentials per second. A hammer strike to that same area will cause that exact same neuron to fire frantically up to 1,000 times a second.
Oh, I see. The brain translates the tempo, the rapid-fire frequency of the signal, as an increase in intensity.
Exactly. And the second strategy is population coding, which makes intuitive physical sense. A gentle touch with a feather only depresses a tiny surface area of skin, activating just a handful of sensory neurons.
But a hammer strike physically crushes and compresses a massive area of tissue, forcefully recruiting hundreds or thousands of surrounding sensory neurons to fire simultaneously.
Yes, the brain listens to the sheer volume of voices shouting at once to determine the severity of the impact.
Okay, so this localized electrical storm travels all the way to the end of the line, reaching the terminal buttons we mentioned earlier. But here, the nervous system encounters a massive logistical hurdle. The terminal buttons of the sending neuron do not physically touch the dendrites of the receiving neuron.
No, there is a microscopic fluid-filled gap between them called the synaptic gap, or the synapse. And the electrical action potential, for all its speed, cannot jump across empty space.
The electrical signal hits a literal dead end. So to pass the message across the void, the communication has to completely change languages. It must translate from an electrical spark into a chemical courier.
This translation process is called synaptic transmission, and it is the physical foundation of all learning, memory, and complex brain function. Inside the terminal buttons, docked near the membrane, are thousands of tiny spherical sacs called synaptic vesicles.
You can visualize them as microscopic water balloons, but instead of water, they're filled with thousands of molecules of specific chemicals known as neurotransmitters.
When the electrical action potential crashes into the terminal button, the change in voltage forces these vesicles to merge with the outer cell membrane. They burst open, vomiting their neurotransmitter cargo into the fluid of the synaptic gap.
These chemical couriers float across the incredibly narrow gap and bump into the dendrites of the receiving neuron. And this interaction is highly specific. It operates on a strict lock and key mechanism.
Right. The neurotransmitter molecule is a key with a specific three-dimensional physical shape. The receiving dendrite is covered in receptor sites, which act as locks. A neurotransmitter can only bind to a receptor site that is perfectly molded to fit its unique shape.
And when that chemical key slots perfectly into the receptor lock, it causes the ion channels on the receiving neuron to open or close, altering its internal voltage. And this is crucial for understanding how the brain processes information. The neurotransmitter does not simply tell the next neuron, fire.
It is far more democratic and mathematical than that. Some neurotransmitters carry an excitatory message. When they bind, they open channels that allow positive sodium ions to leak into the receiving cell. This nudges the internal voltage from -70 slightly closer to the -55 millivolt firing threshold. They are casting a vote that says, yes, you should fire.
But other neurotransmitters carry an inhibitory message. When they bind, they open completely different channels that allow positive potassium to leak out or negative chloride to flow in. This drives the internal voltage down to -75 or -80 millivolts.
It makes the cell hyperpolarized, pushing it further away from the firing threshold. These neurotransmitters are casting a vote that says, no, stay quiet, do not fire.
And in the chaotic environment of the brain, a single neuron might have 10,000 synapses connecting it to thousands of other neurons. At any given millisecond, it's being bombarded by a mixture of excitatory and inhibitory neurotransmitters.
The receiving cell constantly summates. It mathematically tallies all of these competing signals. If the total accumulation of yes votes outweighs the no votes enough to push the voltage to -55, the neuron generates its own action potential, and the cycle continues.
If the inhibitory signals dominate, the neuron remains silent and the message stops there. It's a microscopic, high-speed biological democracy occurring trillions of times a second.
But there is a cleanup problem. Once the chemical key has turned the lock and delivered its vote, it can't just stay in the receptor forever, or the channel would be permanently stuck open.
The synaptic gap must be rapidly cleared so the system can receive the next message. The brain utilizes two primary cleanup crews. The first is reuptake. The sending neuron basically acts as a chemical vacuum cleaner, sucking the neurotransmitters back up out of the gap, repackaging them, and recycling them for later use.
The second method is degradation, where specialized enzymes floating in the synapse act like biological shredders, physically breaking the neurotransmitter molecules apart so they can't bind anymore.
To ground this highly abstract cellular process in reality, we need to look at the specific neurotransmitters themselves. This is where microscopic chemistry dictates our macroscopic psychology, and where modern pharmacology intervenes when things go wrong. Let's start with acetylcholine, commonly abbreviated as A.
ACEH is a major player with two incredibly distinct day jobs. Inside the brain, particularly in networks involving the hippocampus, it is absolutely vital for attention and the formation of new memories. The tragic cognitive decline seen in Alzheimer's disease is directly linked to the massive degeneration of the specific neurons that produce acetylcholine.
But outside the brain, ACA has a completely different function. It's the sole neurotransmitter released at every single neuromuscular junction. That means every time a motor neuron connects to a skeletal muscle, it dumps AC into the gap to tell the muscle fibers to contract.
If you wiggle your fingers right now, that is acetylcholine bridging the gap, which implies a terrifying vulnerability. If you interfere with ACEs, you don't just mess with memory, you completely disrupt physical movement.
That vulnerability is exactly what some of the most potent neurotoxins in the natural world exploit. Consider botulinum toxin, which is produced by bacteria often found in improperly sealed canned food. This toxin prevents the sending neurons from releasing He into the synapse.
So the brain sends the electrical command to move, but the chemical messenger is trapped. The muscle never gets the signal, resulting in total flaccid paralysis, which typically causes death by suffocating the respiratory muscles.
We actually weaponize a purified microdose version of this deadly toxin, cosmetically Botox, to deliberately paralyze facial muscles and smooth out wrinkles. It is wild to think about injecting a paralyzing agent into your forehead for aesthetics.
On the terrifying flip side of the coin, certain synthetic nerve gases developed for chemical warfare operate on the cleanup crew. They destroy the degradation enzymes that are supposed to chew up He.
So the motor neuron fires once, but the acetylcholine floods the synapse and just stays constantly bombarding the receptors. This causes the muscles to lock up in violent, uncontrolled, continuous spasms. So whether you block the release or block the cleanup, disrupting ACH is lethal.
Let's shift to a pair of neurotransmitters that are chemically quite similar but drive vastly different psychological experiences: norepinephrine and dopamine. Norepinephrine is primarily the brain's alertness and physiological arousal chemical. It dictates your baseline mood and readiness to react.
When someone takes a stimulant like cocaine or amphetamines, those drugs act primarily by shutting down the reuptake vacuums for norepinephrine. The chemical is released naturally, but because the vacuum is broken, it accumulates massively in the synaptic gap, constantly overstimulating the receiving neurons and artificially inducing a state of intense manic arousal.
And dopamine is the chemical most famously associated with reward and pleasure. But the modern research refines that definition beautifully. Dopamine isn't just about experiencing pleasure. It heavily regulates what addiction researchers call incentive salience.
It dictates motivation. It dictates how desperately you want to pursue a reward, whether that reward is a sugary donut, a hit of a drug, or the little red notification bubble on your social media app. It drives the behavior to seek the reward.
But the architecture relying on dopamine is staggeringly fragile. A surplus of dopamine activity in certain cortical areas is one of the primary drivers of the auditory hallucinations, paranoia, and fragmented reality seen in schizophrenia. The most effective antipsychotic medications we have developed work as antagonists. They physically plug up and block the dopamine receptors to reduce the chemical's influence.
But it's a tightrope walk, because if you reduce dopamine too much, or if the dopamine-producing neurons in the midbrain naturally die off, you cross into entirely different pathology: Parkinson's disease. Patients with Parkinson's suffer from severe physical tremors, muscle rigidity, and a profound difficulty initiating voluntary movement.
It is terrifying to realize that you have a single chemical where an excess can fracture your perception of reality and a deficit can physically freeze your body in place. We are balancing on a microscopic chemical razor blade. We see a similar balance with serotonin.
Serotonin is heavily involved in regulating baseline mood, sleep cycles, and appetite. Chronically low levels of serotonin are strongly correlated with clinical depression. This correlation led to the development of SSRI's selective serotonin reuptake inhibitors, which are the most widely prescribed antidepressants globally, including drugs like Prozac and Zoloft.
Just as cocaine breaks the vacuum for norepinephrine, SSRIs selectively block the reuptake pumps for serotonin, leaving the molecule in the synaptic gap longer to continually stimulate the receptors and hopefully elevate the patient's mood.
And fascinatingly, hallucinogens like LSD exert their profound reality-bending effects because their molecular structure acts as a master key that perfectly fits and overactivates specific serotonin receptors across the cortex.
Finally, we have to mention the brain's primary traffic control system, glutamate and GABA. They aren't as famous as dopamine, but they do the heavy lifting. Glutamate is the brain's main green light. It is the most abundant excitatory neurotransmitter, constantly nudging neurons to fire.
It is deeply involved in strengthening synaptic connections during learning, heavily utilizing a specific receptor site known as the NMDA receptor. If glutamate is the accelerator, GABA is the heavy brake pedal. GABA is the major inhibitory neurotransmitter. It floods the system with no votes, hyperpolarizing cells, and calming the electrical storm down.
And maintaining the balance between glutamate and GABA is the difference between a functioning mind and a catastrophic seizure. If GABA function is artificially blocked, the excitatory signals spiral out of control, causing severe convulsions. Conversely, clinical anti-anxiety medications, the benzodiazepines like Valium or Xanax, operate by enhancing the efficiency of GABA. They help the brake pedal depress more easily, broadly dampening neural activity and inducing a state of pharmacological calm.
I have to pause here because synthesizing all of this induces a very specific kind of existential dread for me. When you break it down to this level of mechanics, human agency feels incredibly tenuous. I like to believe that my good mood today is because I have a positive outlook on life, or my sudden anxiety is a rational response to my environment.
But you were telling me that I am entirely at the mercy of microscopic chemical soups, a slightly malfunctioning reuptake pump, or a degradation enzyme that is 5% too efficient, and my entire perception of reality, my personality, my physical ability to walk across the room drastically alters. We are profoundly fragile. We are delicate, yes, but we must also recognize that this system is incredibly robust and highly organized. These neurotransmitters are not just sloshing around randomly in your skull like soup in a bowl. They are synthesized, regulated, and pumped through highly structured, specific geographical networks within the brain. To understand how this chemical democracy actually results in complex behavior, we have to zoom out from the cellular level and map the macrogeography of the brain itself.
To navigate the brain, our sources mention a historical model proposed by Paul MacLean, known as the triune brain. MacLean suggested looking at the brain as three concentric layers built sequentially by evolution over millions of years. An ancient primitive reptilian core at the bottom handling survival, a middle mammalian layer handling emotion, and a massive new primate layer wrapped around the outside handling rational thought.
It is conceptually elegant, but modern anatomists prefer a stricter geographic model, dividing the brain from the bottom up into the hindbrain, the midbrain, and the forebrain.
I like to visualize the brain as a sprawling ancient city that simply never stopped expanding. The oldest, most critical plumbing, the foundational life support systems are buried deep at the bottom and the center. And as evolution progressed, newer, more sophisticated architectural layers were just built up and over the old foundations.
That architectural analogy tracks perfectly with the anatomy. Let's start deep in the basement, right where the spinal cord swells as it enters the base of the skull. This is the hindbrain. This region houses the non-negotiable tools for baseline biological survival. The lowest structure is the medulla.
It's a small stalk that manages the absolute basics: the autonomic regulation of heart rate, blood pressure, and the rhythmic expansion of your lungs. Damage the medulla, and the machinery simply turns off.
Just above it sits the pons, which acts as a major physical bridge for nerve fibers crossing from one side of the brain to the other, and houses clusters of neurons involved in regulating sleep stages and physical attentiveness. But the most prominent feature of the hindbrain sits tucked into the back of the skull, the cerebellum.
The cerebellum translates from Latin as the little brain, and it literally looks like one. It looks like a separate, deeply wrinkled miniature brain clinging to the back of the brainstem.
It does, and despite its size, it holds over half the neurons in the entire central nervous system. The cerebellum is the supreme master of physical coordination, timing, and motor learning. If the higher regions of your brain make the conscious decision to pick up a glass of water, they send a sloppy general command. It is the cerebellum that instantly calculates the complex subconscious trigonometry required to contract the bicep, extend the tricep, and steady the shoulder simultaneously so the movement is fluid and precise.
If the cerebellum is damaged by a stroke, the patient doesn't lose the ability to move, but their movements become incredibly jerky, exaggerated, and severely uncoordinated. Moving up from the basement, we reach the midbrain. In humans, it is relatively small, completely dwarfed, and surrounded by the massive structures above it.
It contains important relay stations for processing auditory and visual reflexes, like automatically turning your head when you hear a loud crack. It also houses the substantia nigra, which is that critical dopamine-producing factory that breaks down in Parkinson's disease. But perhaps the most conceptually fascinating structure passing through the midbrain is a diffuse network of interconnected neurons called the reticular formation.
The reticular formation extends down into the hindbrain and stretches up toward the forebrain. It acts as the brain's ultimate bouncer and sensory filter. It is the gatekeeper of consciousness. Millions of sensory signals from your peripheral nervous system hit the reticular formation every second. It evaluates them and decides which signals are important enough to pass up to your conscious awareness and which signals should be ignored so you don't go insane from sensory overload.
The sources cite a famous experiment from the 1940s that highlights just how powerful this bouncer is. Researchers carefully implanted microelectrodes directly into the reticular formation of a cat. When they applied a specific steady electrical current to the tissue, the cat instantly collapsed into a deep sleep.
But when they adjusted the current to deliver a rapidly changing alerting waveform, a deeply sleeping cat would instantly jolt, fully awake, alert, and scanning the room. They had physically located the literal on-off switch for mammalian consciousness. It explains why damage to this specific network, like from a severe concussion, often results in a permanent coma. The hardware above it might be perfectly fine, but the power switch is broken in the off position.
It is a sobering realization. Now, as we move above the midbrain, we encounter the massive canopy that defines human intelligence, the forebrain. This region is massive and is divided into the subcortical structures, meaning the hubs hidden underneath the surface and the wrinkled outer layer, the cortex. Let's explore the subcortical hubs first. Sitting directly atop the brainstem, right in the center of the brain, is the thalamus.
Think of the thalamus as the central post office or the main sensory relay station. With the notable exception of smell, every single piece of sensory information, every photon hitting your eye, every sound wave vibrating your ear, every touch on your skin must stop at the thalamus first. The thalamus receives the data, organizes it, and routes it to the correct specialized processing center in the cortex above.
Tucked just beneath the thalamus is a structure roughly the size of a kidney bean, the hypothalamus. It is tiny, but it punches wildly above its weight class. It is the ultimate thermostat for the entire body. It is dedicated to maintaining homeostasis, keeping the biological environment perfectly stable.
If your internal temperature drops, the hypothalamus detects it and sends signals to make you shiver to generate heat. If your blood sugar drops, it creates the psychological sensation of hunger to force you to eat. Alongside managing survival drives like thirst and reproduction, it also houses incredibly potent reward centers that reinforce those survival behaviors with intense pleasure.
And closely interacting with the hypothalamus is a collection of ancient structures known as the limbic system. This interconnected ring forms the emotional engine of the mammalian brain. If you look at a reptile, its behavior is heavily rigid and instinct-driven. It eats, it fights, it mates. But the evolution of the limbic system in early mammals imposed a necessary layer of emotional memory and behavioral flexibility over those raw instincts. Two structures within the system are paramount: the hippocampus and the amygdala.
The hippocampus is the archivist. It is utterly critical for the consolidation of new explicit memories, taking the events of your day and writing them into long-term storage. And we understand its function with absolute certainty because of a profoundly tragic era in medical history. In the 1950s, a man known in the literature as HM suffered from debilitating, life-threatening epileptic seizures. Desperate for a cure, a surgeon performed an experimental and aggressive procedure, physically excising HM's left and right hippocampi.
The surgery successfully stopped the seizures, but it resulted in a catastrophic cognitive deficit. By removing the hippocampi, the surgeon permanently destroyed HM's ability to form any new memories.
And it wasn't that his intelligence was damaged. If you met HM, he was articulate, he had his full vocabulary, and he perfectly remembered his childhood from before the surgery. You could sit and have a fascinating conversation with him for an hour. But if you briefly stepped out of the room to get a cup of coffee and walked back in, HM would look at you as a total stranger. He would politely introduce himself again. He was permanently, inextricably anchored in the present tense, completely unable to carry a new fact forward in time. His tragedy fundamentally rewrote our understanding of how memory is geographically localized in the brain.
Just adjacent to the hippocampus is the amygdala, a small, almond-shaped cluster of nuclei that acts as our emotional sentinel, specifically tuned to threats. The amygdala is the primary hardware for fear conditioning. We know from lesion studies that if a monkey's amygdala is surgically damaged, the animal entirely loses its natural fear of predators, like snakes, and it loses its wariness of aggressive dominant peers. It becomes dangerously fearless. In human patients with amygdala damage, they lose the ability to accurately recognize the expression of fear on another person's face, and they struggle immensely to learn to associate a neutral stimulus with a painful outcome.
It's the brain's alarm bell. But what I find most fascinating is how the amygdala interacts with the hippocampus right next door. If you experience something that is emotionally highly charged, a terrifying car accident, or an incredibly thrilling first kiss, the amygdala fires aggressively. It essentially screams at the hippocampus, "This moment is vital for survival. Burn this memory deep." This is why you vividly remember where you were during a major global tragedy, but you have zero memory of what you ate for lunch last Tuesday. The amygdala didn't care about Tuesday's lunch, so the hippocampus didn't bother saving it.
And recent sociocognitive research even suggests that the physical volume of a person's amygdala positively correlates with the size and complexity of their active social network, hinting that managing the emotional nuances of many relationships requires serious hardware. Now, all of these subcortical systems, the medulla, the thalamus, the limbic engine, are continuously running, but they operate beneath the surface of conscious thought. To finally reach the seat of human consciousness, to reach the architecture that allows us to ponder our own existence, we have to travel up to the convoluted outer rim of the city. We have to explore the cerebral cortex.
The word cortex literally comes from the Latin word for the bark of a tree, which makes visual sense. It is the outermost layer of the brain, a heavily folded sheet of tissue only about 2 to 3 millimeters thick. It is completely wrinkled and convoluted into deep grooves and ridges, giving the brain that classic walnut-like appearance. The brain does this to cheat physical geometry. Folding the tissue allows nature to pack a massive amount of surface area into the small, limited volume of the human skull. And because the cortex is primarily composed of the unmyelinated cell bodies of neurons, it appears grayish to the naked eye, which is why we colloquially call intelligence using your gray matter. The bright white myelin insulation on the axons carrying signals away from the cortex forms the massive web of white matter beneath it.
Structurally, the cerebral cortex is split squarely down the middle into two distinct halves, the left hemisphere and the right hemisphere. These two halves communicate continuously across a massive, dense bridge of millions of nerve fibers called the corpus callosum. Anatomists further divide each hemisphere geographically into 4 distinct lobes. You have the frontal lobe situated behind the forehead, the parietal lobe stretching across the top rear, the occipital lobe tucked all the way at the lower back of the skull, and the temporal lobe sitting on the sides roughly behind your ears.
And mapping what happens in each of these lobes is where neuroscience gets incredibly specific. Let's look at the strip of tissue sitting at the very front of the parietal lobe. This is the primary somatosensory cortex. This specific strip is dedicated entirely to receiving tactile information from the body touch, temperature, pressure, pain. Immediately in front of that, sitting at the back edge of the frontal lobe, is the primary motor cortex. This strip is the outbound command center, responsible for executing all voluntary muscular movement.
The mapping of how the physical body connects to these two strips of cortex is one of the most famous visual models in neuroscience. It is called the homunculus, which translates to little man. In the 1940s, a pioneering Canadian neurosurgeon named Wilder Penfield mapped this geography through direct physical experimentation.
This is one of my favorite stories. Penfield was performing open brain surgery on conscious patients to treat severe epilepsy. Now, because the brain tissue itself has absolutely no pain receptors, a patient can remain wide awake and communicative while the surgeon is probing their cortex. Penfield would take a tiny electrical probe, apply a minute electrical current to a highly specific spot on the motor cortex, and simply watch what part of the patient's body twitched. By doing this hundreds of times, he drew a precise map of the body on the surface of the brain.
Penfield's map revealed 2 deeply counterintuitive facts. First, the geography is inverted. If Penfield stimulated the very top of the motor cortex near the center part in your hair, the patient's toes would involuntarily wiggle. If he stimulated the tissue way down at the bottom of the strip near the ear, the patient's tongue or jaw would involuntarily move. The body is mapped upside down on the brain.
And the second discovery was that the map is wildly, almost comically distorted. The amount of physical brain space dedicated to a body part has absolutely no correlation to the physical size of that body part. If you built a physical model of a human being where the size of the body parts matched the amount of cortical real estate they were assigned, you would create a grotesque monster. The homunculus has a tiny, spindly little torso and tiny stick legs because your back and legs don't do complex movements. They mostly just keep you upright. But this monster would have massively swollen gigantic hands with enormous fingers and incredibly huge lips, tongue, and jaw.
It perfectly illustrates biological economics.
Mhm.
Cortical real estate is incredibly expensive biologically. Nature does not assign brain space based on physical size. It assigns space based on the sheer precision of motor control required for survival. Your hands require massive cortical computing power to execute the incredibly delicate fine motor skills needed to fashion a tool, thread a needle, or play a violin. Your lips, tongue, and vocal cords require phenomenal neurological coordination to instantly shape the rapidly changing puffs of air that create complex, articulate speech. Function dictates form.
And you see this exact economic logic when you map the cortex of animals. If you map a dog's motor cortex, it allocates very little tissue to its front paws because a dog uses its paws primarily just for walking and running gross motor skills. But if you map the brain of a raccoon, an animal that relies entirely on using its highly sensitive paws to explore the environment, pry things open, and watch its food, you find a massive swath of cortex dedicated entirely to its forepaws and even individual digits. Or consider a rat. A rat navigates dark, subterranean environments almost entirely by feeling its way with its whiskers. When you map a rat's sensory cortex, you literally find a dedicated, distinct patch of neural tissue for every single individual whisker on its face. The brain perfectly molds itself to the survival strategy of the organism.
Precisely. Now, moving away from touch and movement, the other lobes house distinct sensory processors. The occipital lobe at the very back of your head is dedicated exclusively to the complex processing of visual information. And vision features a fascinating anatomical quirk regarding how the wires cross. The optic nerves extending from the inner half of each retina cross over each other at an intersection called the optic chiasm. Because of this crossing, all the visual information originating in the left half of your visual field is routed across the brain to be processed by your right hemisphere, and everything in your right visual field is routed to the left hemisphere.
Similarly, the temporal lobes on the sides of your head contain the primary auditory cortex for processing sound. But if you map out all these primary sensory and motor strips, they only account for a small fraction of the total human cortex. The vast majority of the tissue, roughly 75%, is made up of what we call association areas. And this is where raw sensory data is woven together to create complex thought. This is where the magic of human cognition lives.
The most critical and highly developed association area in humans is the prefrontal cortex, situated at the very front of the frontal lobes right behind your forehead. You can think of the prefrontal cortex as the CEO of the brain. It doesn't process raw vision or command a specific muscle to twitch. Instead, it handles executive function. It is responsible for long-term planning, moral reasoning, complex decision-making, and perhaps most importantly, impulse control. It actively inhibits the primal urges bubbling up from the limbic system.
And we know this because what happens when the CEO is fired. If a patient suffers a severe traumatic injury to the prefrontal cortex, they don't go blind and they don't become paralyzed. Their basic biological functions are fine, but their personality can completely unravel. They may lose all ability to plan for the future, becoming highly impulsive. They might lose their social filter, acting wildly inappropriately in public because the hardware that normally suppresses those behaviors have been destroyed. They lose the rational control over the animal machine. We also have posterior association areas near the sensory maps. The sources describe a condition where a patient suffers damage to the visual association area in the lower temporal lobe. They develop a bizarre deficit called visual agnosia. If you hand them a drawing of a bicycle, their eyes work perfectly. They can trace the shape with a pencil, they can describe the curves and the straight lines, but they cannot tell you what the object is. The raw visual data is arriving cleanly, but the association area that links that visual shape to the semantic meaning of bicycle is broken.
Now, you brought up Wilder Penfield physically poking exposed brains with electrodes to figure all this out. For a long time, that was our only methodology, relying on gruesome accidents, studying the behavioral deficits of stroke victims, or surgically lesioning the brains of animals. But the technological revolution of the late 20th century provided us with an incredible suite of non-invasive neuroimaging tools. We can finally peer inside the living, working human skull.
It is a massive leap forward, and we generally divide these tools into two categories. First, we have structural imaging, which takes high-resolution photographs of the brain's physical architecture. We use CT or CGAS scans, which take multiple X-rays from different angles and use a computer to build a 3D image. And we have MRI, magnetic resonance imaging, which uses incredibly powerful magnetic fields and radio waves to map the density of tissue. If you're looking for a physical tumor, a structural deformity, or bleeding in the brain, an MRI provides a pristine map. But an MRI is just a static photograph. It tells you nothing about what the brain is actually doing.
For that, we turn to functional imaging. These tools allow us to watch the mind in action in real time. PET scans, positron emission tomography, involve injecting the patient with a mildly radioactive tracer attached to glucose. Because active, hard-working neurons consume more glucose for energy, the PET scanner can track the radiation and generate a color-coded map showing exactly which areas of the brain light up while the patient reads a book or solves a math problem. Even more prominent today is fMRI, functional MRI. Instead of radiation, fMRI tracks the magnetic resonance of oxygenated blood. When a brain region suddenly works harder, it demands more oxygen and blood rushes to that specific area. fMRI tracks these hemodynamic changes with incredible spatial precision and across very tight time windows.
And this functional imaging isn't just for finding disease. It allows us to answer mysteries of human performance that seem biologically impossible. Here's one of my favorite applications from the sources, looking at the cutting edge of sports psychology. The expert athlete. Imagine a professional tennis player standing on the court, preparing to return a serve that is traveling at 150 miles per hour. From a purely biological perspective, returning that serve should be impossible. Why? Because the human nervous system has a strict, hardwired speed limit. It takes roughly 100 milliseconds for the physical light of the ball to hit the retina, turn into an electrical signal, and travel back to the occipital lobe. And it takes at least another 100 milliseconds for the frontal cortex to decide on a swing, route the command down the spinal cord, and cause the arm muscles to contract.
That irreducible 200 millisecond biological lag means that human beings are essentially living a quarter of a second in the past. If the tennis player waits to consciously see the ball leave the racket before they initiate their reaction, the ball will physically blow past them before their arm can even begin to move. So the question is, how do they consistently hit it?
They do it by predicting the future to bypass the biological delay. Researchers studying expertise like Anders Ericsson argue that elite athletes don't just have faster reflexes. They achieve their performance through thousands of hours of grueling, deliberate practice. They don't just play for fun, they intensely practice specific, difficult scenarios that force the brain to adapt. This brutal repetition fundamentally rewires their perceptual networks to read advanced cues. The pro tennis player isn't actually waiting to see the ball. Their brain is calculating the subtle drop of the server's shoulder, the rotation of the hips, and the exact angle of the racket face milliseconds before the impact.
Yeah. And fMRI scans vividly illustrate this neurological superpower. If you place a world-class tennis player inside a scanner and have them watch videos of serves, their functional brain network looks fundamentally different from an amateur's. The amateur's brain shows high activity in basic visual and frantic motor planning areas, but the expert's brain shows intense activation in highly specialized motor imagery and spatial prediction networks. The expert is subconsciously running a hyperfast physics simulation in their parietal and frontal lobes, feeling the trajectory of the ball and initiating the muscle commands before the event even officially occurs. They have rewired their wetware to outsmart the speed biology.
It is the closest thing to actual magic in biology. Now, while functional imaging showed us how experts use their whole brain differently, it also definitively proved a massive anatomical quirk that we touched on earlier. When you look at a photograph of the brain from the top down, the left hemisphere and the right hemisphere look like perfect mirror images. They look like identical twins. But functionally, they run entirely different operating systems.
This phenomenon is known as lateralization, or hemispheric specialization. It is the realization that certain cognitive functions are heavily localized to one side of the brain. The most prominent and historically significant example of this is human language. The medical community first stumbled upon this asymmetrical design in the 1860s through the study of aphasia, the loss of the ability to produce or understand language due to brain damage. A French surgeon named Paul Broca encountered a patient who was cognitively intact but had completely lost the physical ability to speak except for a single repetitive syllable, tam. When the patient eventually died, Broca performed an autopsy and found a massive localized lesion in the left frontal lobe.
That specific region became known as Broca's area, and it is the critical hub for speech production. Patients who suffer a stroke in this area develop expressive aphasia. The tragic irony of this condition is that the patient knows exactly what they want to say. Their internal thoughts are clear. But they speak in slow, agonizing, telegraphic bursts, struggling immensely to physically coordinate the words walk, dog, now. The grammatical structure is destroyed, but the meaning is there.
Roughly a decade later, a German neurologist named Karl Rönecke discovered a completely different form of aphasia linked to a lesion further back in the left temporal lobe. These patients suffered from receptive aphasia. Their speech was completely fluent. They spoke at normal speed with perfect rhythm, articulation, and grammar. But the sentences they produced were complete, incomprehensible gibberish. They would invent words and string unrelated concepts together in a word salad. Furthermore, they entirely lost the ability to comprehend spoken or written language from others. The physical ability to articulate was intact, but the neurological dictionary that assigns meaning to sound had been destroyed.
In the 20th century, a researcher named Norman Geschwind synthesized these findings into a unified pathway called the Wernicke-Geschwind model. It elegantly maps exactly how a thought moves through the brain to become speech. If you are reading a word aloud, the raw visual data hits the visual cortex. It is routed to the angular gyrus, which translates the visual shape into an auditory code. That code is sent to Wernicke's area, which searches the database to comprehend the meaning of the word. Wernicke's area then transmits that meaning forward to Broca's area, which drafts the highly complex motor plan required to speak the word. Finally, Broca's area sends the finalized blueprint to the motor cortex, which fires the exact sequence of muscles in your lips, jaw, and vocal cords. If any single node or pathway in that relay is severed, a highly specific element of language breaks down.
And here is the crucial point regarding lateralization. In the vast majority of the human population, nearly 95% of right-handed people, this entire complex language machinery, Broca's area, Wernicke's area, the angular gyrus, is housed exclusively in the left hemisphere. The corresponding regions in the right hemisphere are largely mute. They handle rhythm, intonation, and metaphor, but not the mechanical processing of syntax and vocabulary.
The left brain is the talking brain. But to truly grasp the profound implications of this division, we have to examine the Nobel Prize-winning research of Roger Sperry in his work with split-brain patients. We mentioned earlier that the two hemispheres constantly communicate across a massive cable of nerve fibers called the corpus callosum. In patients suffering from severe drug-resistant epilepsy, a violent electrical seizure can originate in one hemisphere, travel across the corpus callosum like a bridge, and ignite the other hemisphere, causing a catastrophic full-body grand mal seizure.
In the mid-20th century, neurosurgeons devised a radical last-resort intervention. To trap the electrical storm on one side of the brain, they surgically severed the corpus callosum. They physically cut the bridge, completely disconnecting the left hemisphere's cortex from the right hemisphere's cortex. In their everyday lives, these split-brain patients seemed remarkably unaffected. They walked, talked, and reasoned normally. But Sperry realized that under highly controlled laboratory conditions, he could expose the fact that these patients now harbored two isolated, disconnected streams of consciousness in one skull.
Sperry's experiments relied on the optic chiasm crossing we discussed earlier. He had the patient sit down and stare directly at a small dot in the exact center of a screen. Because the eyes are locked on the center, anything flashed incredibly quickly to the left of the dot is processed exclusively by the right hemisphere. Anything flashed to the right of the dot goes exclusively to the left hemisphere. And remember, the motor nerves cross over too. The left hemisphere controls the right hand, and the right hemisphere controls the left hand.
This anatomical setup allowed Sperry to interrogate one half of the brain without the other half knowing.
Right, so Sperry hides the patient's hands behind a screen so they can't see them. He flashes the word nut-t on the left side of the screen for a fraction of a second. That visual information travels exclusively to the right hemisphere. Sperry then asks the patient, "What did you just see?" Now, the speech center, Broca's area, is trapped in the left hemisphere. The left hemisphere was looking at the blank right side of the screen. It genuinely saw nothing. So the patient verbally replies, "I didn't see anything."
The conscious speaking self is completely unaware of the word.
But then Sperry asks the patient to use their left hand, which is controlled by the right hemisphere, the side that did see the word, to reach under the screen and feel around a pile of hidden objects. Without the aid of vision, the left hand unerringly searches the pile, finds the nut, and holds it up. It is a staggering scene. The patient is verbally denying seeing anything, swearing the screen was blank, while their own left hand is actively holding up the evidence. The right hemisphere knows exactly what it saw, and it has the physical intelligence to find it, but it is entirely mute. It cannot speak its truth to the left hemisphere.
The isolation becomes even more surreal when Sperry fed conflicting information to both sides simultaneously. In one experiment, he flashed a compound word across the center dot: hat band. The word hat lands in the left visual field, going to the right brain. The word band lands in the right visual field, going to the left brain. Sperry asks the patient, "What word did you see?" The left brain, possessing the power of speech, immediately says, band. Sperry then asks, what kind of band? And because the left brain has no access to the word hat, the patient begins wildly guessing, a rock band, a rubber band. The left hemisphere, desperate to maintain a cohesive narrative of reality, simply fabricates a story to cover up the missing information.
There are accounts where the disconnection leads to literal physical conflict between the limbs. If you ask a split-brain patient to arrange a set of colored blocks into a complex geometric pattern, the right hemisphere, which excels at spatial reasoning and geometry, can easily command the left hand to complete the puzzle. But if the patient tries to use their right hand, driven by the spatially clumsy left hemisphere, they struggle miserably. In some documented cases, as the right hand stumbles and makes mistakes, the left hand will literally shoot out, swat the right hand away, and fix the puzzle itself. One brain physically fighting itself over a task.
It forces us to ask terrifying questions about the unity of the self. However, it is paramount to clarify that in a neurotypical brain with an intact corpus callosum, you do not have two independent minds fighting for the steering wheel. The connection is instantaneous and flawless. The brain integrates the specialized talents of both sides. When you read a novel, your left hemisphere is doing the mechanical heavy lifting, decoding the syntax, retrieving the dictionary definitions of the words. Simultaneously, the right hemisphere is processing the emotional tone of the scene, interpreting the metaphors, and understanding the subtle humor. It requires both halves working in perfect unison to experience the full depth of the narrative. It is a completely unified, singular experience.
This deep dive into localized functions like Broca's area and the split brain brings us squarely into one of the most intense ongoing debates in the history of neuroscience. The debate centers on a simple question. Is the brain a collection of highly specialized, isolated factories doing specific jobs, or is it one giant, globally interconnected web where everything does a little bit of everything?
This tension dates back to the early 19th century and the bizarre pseudoscience of phrenology. A physician named Franz Joseph Gall aggressively championed the idea of extreme localization. He proposed that highly specific human traits, things like vanity, combativeness, or the memory of facts, were localized in very specific, distinct regions of the cortex. Gall argued that if a person exercised a trait, like vanity, that specific piece of brain tissue would physically grow, pushing outward against the skull and creating a palpable bump. Phrenologists would literally run their hands over a patient's head and read their personality based on the lumps.
Which is obvious quackery today. But the core theoretical assumption beneath the quackery, the idea that different geographic areas handle different functions, was partially validated later by Broca and Wernicke finding language centers and Penfield drawing the motor map. For a long time, neuroscience was obsessed with strict functional specialization. We wanted to point to a millimeter of tissue and say, that is where jealousy lives.
However, modern neuroscience has heavily shifted away from extreme localization toward a paradigm called connectionism, heavily utilizing complex mathematics like graph theory. Connectionists argue that while specialized hubs certainly exist, it is a mistake to think of them as isolated factories. In graph theory, the brain is modeled as a massive network. The physical regions of tissue are called nodes, and the white matter pathways connecting them are called edges. The focus has shifted from asking where is the language center to asking how efficiently do all the diverse nodes communicate globally to produce a thought.
And this shift isn't just academic. It has profound implications for how we understand devastating mental illness. The sources highlight a fascinating study by Alexander Bloch regarding childhood-onset schizophrenia. Historically, we looked at schizophrenia primarily as a localized chemical problem, too much dopamine in a specific pathway. But when researchers analyzed the brains of these patients using network graph theory, a much deeper structural failure emerged. The entire global network of the brain was fragmented. The researchers found that while the brain was still capable of sending signals across long global distances, the local neighborhood processing groups of nearby nodes trying to collaborate on a complex thought was severely degraded and inefficient.
The fragmentation of the patient's psychological reality, the disordered thoughts, the delusions, was a literal mathematical reflection of the fragmentation of their physical neural network. They had lost the cohesive integration required to maintain a stable reality. Conversely, researchers like Vanden Heuvel have used graph theory to study intelligence in healthy individuals. They put subjects in an fMRI and simply measured the efficiency of their brain's resting state network, how well the nodes communicate when the person isn't even doing a specific task. They found that the overall mathematical efficiency of the global network strongly predicted the person's IQ score.
So it's not about having a physically massive frontal lobe. Intelligence is a measure of bandwidth. It is a measure of how efficiently the disparate regions of the brain can seamlessly collaborate. So to reconcile the historic debate, the brain is neither strictly localized nor entirely global. It operates through specialized hubs embedded within a highly flexible plastic web that constantly shifts its connections based on learning and demand.
That is the modern consensus. Now, we have spent almost this entire time examining the rapid, high-speed electrical communication of the nervous system. But the brain does not operate in a vacuum. It is deeply influenced and often controlled by slower, broader chemical operators running in the background. We need to circle back to the autonomic nervous system we mentioned earlier and introduce its powerful biochemical partner, the endocrine system.
Let's start with the autonomic system, the biological autopilot controlling the internal organs. This system manages homeostasis by constantly balancing two opposing teams. The first team is the sympathetic nervous system. This is the biological accelerator pedal. It activates heavily during emergencies, intense stress, or extreme arousal. It initiates the famous fight-or-flight response. If you are hiking and a bear suddenly steps onto the trail, your sympathetic system instantly takes over. It dilates your pupils to let in maximum light, it accelerates your heart rate to furiously pump oxygen to your skeletal muscles, and it completely shuts down non-essential systems like digestion. Logic is brutal. You don't need to waste energy digesting a sandwich if you are about to be eaten.
And its antagonist is the parasympathetic nervous system. This is the heavy brake pedal. Once the bear wanders away and the threat passes, the parasympathetic system kicks in to restore baseline calm. It is colloquially known as the rest and digest system. It constricts the pupils, slows the pounding heart, and routes blood flow back to the stomach and intestines to resume digestion and energy storage. The dynamic, seesawing balance between these two systems keeps your internal environment stable.
But the autonomic system achieves its most profound body-wide effects by partnering with the endocrine system. I like this analogy. If the nervous system operates like an instantaneous email, firing highly targeted electrical signals to specific muscles in milliseconds, the endocrine system is the biological postal service. It is a distributed network of glands that secrete chemical messengers, called hormones, directly into the general bloodstream. These hormones travel slowly through the entire circulatory system, washing over every organ, but they only exert an effect on cells that possess the specific receptor for that hormone. It's a slower system, but the effects are massive and long-lasting.
The absolute CEO of the endocrine system is the pituitary gland. It is a tiny pea-sized structure dangling just below the hypothalamus in the brain. The hypothalamus, acting as the translator between the fast nervous system and the slow endocrine system, tells the pituitary what to do. Let's trace how this partnership handles the stress of the bear on the trail. The brain visually registers the threat. The amygdala sounds the alarm. The hypothalamus immediately secretes a chemical messenger called CRF corticotropin releaser factor down the short stalk to the pituitary gland. The pituitary, receiving the order, releases its own messenger, ACTH, adrenocorticotropic hormone, into the general bloodstream.
That ACTH travels all the way down the torso until it hits the adrenal glands, which sit like little hats on top of your kidneys. The adrenal glands receive the ACTH signal and immediately pump out a cocktail of over 30 different stress hormones to help you survive the encounter. One of the primary hormones is cortisol. Cortisol's job is to rapidly liberate glucose from your fat stores, flooding your blood with emergency energy so your muscles can fight or run. Interestingly, cortisol acts as a double-edged sword for the brain itself. A brief spike of stress, a low level of cortisol, actually sensitizes the hippocampus and enhances memory formation, helping you remember exactly where the bear was so you can avoid it tomorrow.
But chronic stress, a state where the modern human is constantly anxious about deadlines, mortgages, and social media, bathing the brain in high, unrelenting levels of cortisol is deeply toxic. Prolonged exposure to high cortisol literally causes the dendrites in the hippocampus to wither and retract, leading to neural death and severe memory impairment. The system evolved to handle a 5-minute sprint away from a predator, not a 5-year grind in a stressful corporate job.
And while the outer layer of the adrenal gland is pumping out cortisol, the inner core is dumping epinephrine and norepinephrine, commonly known as adrenaline and noradrenaline, into the blood. These hormones act instantly to constrict blood vessels in the stomach, force the heart to beat harder, and dilate the airways in the lungs. Now, wait a minute. You mentioned norepinephrine earlier.
I did.
We just spent a massive amount of time talking about norepinephrine as a neurotransmitter floating across the microscopic synaptic gaps inside the brain to regulate arousal and mood. Now you're telling me it's a hormone being dumped into the bloodstream by the kidneys.
It is the exact same chemical molecule. It is yet another profound example of how frugal nature is with its designs. If the molecule is released by a neuron across a synapse to act instantly on a neighbor, we categorize it as a neurotransmitter. If that exact same molecule is manufactured by a gland and dumped into the blood to slowly influence the entire body, we call it a hormone. Evolution found a chemical key that worked and simply deployed it in two entirely different communication networks.
It is remarkably elegant. So if we take a step back and look at this entire sprawling apparatus we've built, the action potential sparking, the specialized cortical lobes calculating, the global networks fluctuating, the hormonal cascades washing over the body, we are left with the ultimate question. Who wrote the blueprint? Where did the schematic for this incredibly complex biological machine come from? To truly fulfill our mission today, we have to drill down into the ultimate source code: evolution and genetics.
When behavioral biologists analyze why an organism acts the way it does, they divide their explanations into proximate causes and ultimate causes. Let's look at the aggression of male red deer during mating season. If you ask a physiologist, why is that deer aggressively locking antlers with its rival? Well, they will give you the proximate cause. They will point to the immediate internal biological mechanics, because the change in season triggered a massive spike in testosterone, which lowered the threshold for the firing of aggressive neural networks in the hypothalamus.
That explains the how, but it entirely misses the evolutionary why. The ultimate cause asks why nature bothered to select for and build that specific aggressive hardware in the first place. And the brutal answer is reproductive success. Millions of years ago, a male red deer with a docile, peaceful brain architecture would simply back down from a fight. Because he backed down, he failed to secure a harem of females he didn't mate, and his docile genes were permanently deleted from the gene pool. The hyper-aggressive male fought off competitors, mated with multiple females, and passed his aggressive hardware genes to the next generation. Over countless cycles of sexual selection, the biology of the species was fundamentally shaped by whatever traits ensured survival and reproduction. Nature doesn't care about our human concepts of morality or peace. It solely rewards the hardware that passes the code forward.
And that biological code, the blueprint for every neural pathway and neurotransmitter pump, is inscribed in DNA deoxyribonucleic acid. DNA is tightly coiled into microscopic structures called chromosomes, which reside in the nucleus of almost every cell in your body. Every human cell, with the exception of the sex cells, contains 46 chromosomes arranged tightly in 23 pairs. You inherit exactly half of this genetic library from your mother and half from your father. The specific segments of DNA along these chromosomes are the genes. Genes are essentially microscopic instruction manuals. They tell the cellular machinery exactly which proteins to build, dictating everything from your eye color to the density of dopamine receptors in your prefrontal cortex.
And the interaction of these inherited genes dictates our biological destiny through the mechanics of dominant and recessive traits. It's basic genetic math. If you inherit one dominant gene instructing the body to build brown eyes and one recessive gene instructing blue eyes, the dominant instruction always wins. You will have brown eyes. To physically express a recessive trait, you have to be lucky or unlucky enough to inherit two copies of the recessive gene, one from each parent. And this simple genetic math can have devastating, life-altering psychological consequences.
The clearest example of this in the sources is a metabolic disorder called PKU, or phenylketonuria. PKU is caused purely by inheriting two specific mutated recessive genes. If an infant inherits this combination, their liver entirely lacks the ability to produce a specific enzyme needed to digest an amino acid called phenylalanine, which is ubiquitous in common foods like milk, eggs, and bread. If the infant consumes these foods, the undigested amino acid rapidly builds up to toxic levels in the blood. It acts as a poison to the developing central nervous system, aggressively damaging the white matter and causing profound, permanent intellectual disability and behavioral issues.
It sounds like a tragic, inescapable genetic destiny. Your code is broken, so your brain is destroyed. But here is the most incredible, hopeful part of the PKU story. It proves that genetic destiny can be rewritten by the environment. Today, we mandate a simple blood test for every newborn to identify this specific genetic defect. If a child tests positive for PKU, we immediately intervene and place them on a hyper-strict, highly controlled diet that contains absolutely zero phenylalanine. And the result? The genetic defect is still in every cell of their body. The code is still broken. But because we changed the environment, the diet, the poison never builds up, and the child's brain develops with completely normal intelligence. It is the ultimate proof that biology is not a rigid cage. The environment has the power to silence a devastating genetic flaw.
It is a triumph of modern medicine and a perfect illustration of gene-environment interaction. Sadly, not all genetic mandates can be bypassed so easily. Huntington's disease, a horrific neurodegenerative disorder that typically strikes in a person's 30s or 40s, causing uncontrollable movements, severe personality changes, and eventual dementia, is caused by a single dominant gene. If a person inherits just one copy of the Huntington's mutation from either parent, it is a biological certainty that they will develop the disease. The dominant instruction cannot be overridden.
We also see the quirks of genetics in sex-linked traits. The 23rd pair of chromosomes determines a person's biological sex. Females typically inherit 2 large X chromosomes, while males inherit 1 large X and 1 stunted, much smaller Y chromosome. Because a male Y chromosome is essentially missing a lot of genetic information, if a male inherits a recessive, defective gene on his single X chromosome, like the gene that causes red-green color blindness, he is out of luck. He doesn't have a backup X chromosome with a dominant, healthy gene to override it. The defect is immediately expressed. This is why you see color blindness vastly more often in men than in women.
However, when we transition from discussing simple physical traits like eye color or single gene disorders and try to look at complex psychological traits, things like general intelligence, introversion, or emotional volatility, the genetic math becomes exponentially more complicated. Almost all complex human behaviors are polygenic. They are not controlled by a single smart gene or a shy gene. They are the emergent result of hundreds or perhaps thousands of different genes all interacting simultaneously to subtly shape the brain's architecture. And trying to untangle exactly how much of our personality is dictated by this complex polygenic code versus how much is shaped by our upbringing, our parents, and our culture brings us to the oldest battleground in psychology: nature versus nurture. To settle this debate, researchers needed a way to isolate the variables, and nature provided the perfect experimental subjects: twins.
Twin studies are the bedrock of behavioral genetics. You have identical twins, or monozygotic twins, who developed from a single fertilized egg that split. They share 100% of their genetic code. They are biological clones. Then you have fraternal, or dizygotic twins, who develop from 2 separate eggs. They share only 50% of their genes, just like any standard siblings. By comparing these 2 groups, researchers can start to isolate the influence of DNA. But the most powerful data comes from the rare, heartbreaking cases where identical twins were separated at birth and raised in entirely different families, different cultures, and different socioeconomic environments. When you reunite these separated identical twins as adults and test them, the findings are consistently shocking. Identical twins raised completely apart, who have never met, are significantly more similar in their baseline intelligence, their core personality traits, and even their bizarre personal quirks than fraternal twins who were raised together in the exact same household by the exact same parents. Furthermore, when you put these separated identical twins in an MRI machine, the physical structural architecture of their brains, the specific density of their gray matter in the frontal lobes, looks remarkably similar. The underlying biological blueprint exerts an incredibly powerful, undeniable gravity on who they become, regardless of the environment they grew up in.
But the data that truly surprised the scientific community didn't come from twins. It came from massive longitudinal adoption studies. Researchers tracked the cognitive development of adopted children over decades.
Right. I was floored by this finding. When an adopted child is young, say 3 or 4 years old, their cognitive abilities and verbal scores show a moderate correlation with the adoptive parents who are actively raising them, reading to them, and shaping their daily environment. The environment seems to be winning. But as that child ages, going through puberty and approaching 16 or 18 years old, something fascinating happens. The correlation with their adoptive parents fades away, and they become significantly more similar in cognitive traits to their biological parents, people they may have never even met. It completely flips the intuitive script.
Yeah.
You would think the longer a child spends in an environment, the more that environment would shape them. It suggests that genetic influence is not just a static blueprint laid down at birth. Genetic expression is dynamic. As the brain matures, as the prefrontal cortex develops throughout adolescence, new genetic instructions come online, aggressively pushing the individual's cognitive profile toward their biological baseline. The genetic code continues to execute its program over a lifetime. However, and I think this is the most critical caveat to end on, we have to desperately avoid falling into the trap of genetic determinism. Having a specific sequence of DNA does not mean you are a biological robot locked into a specific fate. The sources highlight the research on the D4 dopamine receptor gene. Researchers identified a specific variation of this gene that correlates strongly with novelty-seeking behavior. People with this variation tend to be highly impulsive, quick-tempered, and statistically, they have a significantly higher risk of falling into alcoholism and drug abuse.
And this is the vital nuance: possessing that specific genetic variation does not guarantee that the person will become an alcoholic. A gene simply represents a predisposition, a vulnerability, or a biological potential. It is exactly like diabetes. You might inherit a strong genetic predisposition for type 2 diabetes, but if you eat a pristine diet, exercise rigorously, and maintain a healthy weight, the disease may never physically manifest. The genetic vulnerability requires an environmental trigger like obesity and a high-sugar diet to actualize.
The phrase I love for this is, genetics loads the gun, but the environment pulls the trigger. If a child inherits the D4 novelty-seeking gene, but they are raised in a stable, loving, highly structured environment that channels that impulsive energy into healthy risk-taking, like extreme sports or entrepreneurship, the vulnerability never becomes pathology. The genetic code simply provides the rough clay. The environment still sculpts the final statue.
That gene-environment interaction is the defining synthesis of modern behavioral biology. The two forces are inextricably linked.
All right, let's pull back and look at the massive terrain we have covered today. We started by looking at a medical student who dreamed he was a dog and woke up to find his emotional reality rewritten by a hijacked sensory network. We dug into the microscopic foundations, watching the electrical tension of action potentials leap across insulated gaps, sparking the release of dopamine and serotonin into microscopic democratic chemical elections. We zoomed out to map how those chemical rivers flow through the ancient survival hardware of the limbic system, up into the incredibly specialized yet globally networked geography of the cerebral cortex, where the left and right hemispheres perform a miraculous unified dance. And finally, we realized that this entire staggering biological machine is built from a constantly unfolding blueprint, refined by millions of years of brutal evolutionary trial and error, and inscribed in the very DNA we inherit. We are an unimaginably complex, fragile, highly adapted network of flesh, electricity, and chemistry.
Which leaves us with a philosophical question that neuroscience has yet to fully resolve. If every profound emotion you feel, every memory you cherish of a loved one, and every conscious decision you make is, at its absolute core, the deterministic result of a chain reaction of sodium ions rushing through channels, neurotransmitter votes summating in a synapse, and ancestral genetic code interacting with your environment, where exactly does free will reside in this wet machinery? Is the immaterial soul that Descartes so desperately tried to protect a real entity pulling the levers? Or is our profound sense of conscious self simply a beautiful emergent illusion generated by the humming of 100 trillion synaptic connections?
It is enough to make you genuinely wonder if you are the author of your choices or if your biology is just flawlessly executing its code. Thank you for joining us on this deep dive. Keep questioning the incredible mechanical universe sitting right inside your own skull. And tonight, if you happen to fall asleep and dream that you are a dog, pay very close attention to how the world smells when you wake up.